Hello, my name is Paola Ehrlich,
And I'm Kristy D'Alessandro.
and we will be presenting about BACE1 and BACE2. To start off, we will talk about the background information of BACE1. BACE1 is a transmembrane protein that functions as a dimer. It is mainly found in the brain and pancreas, but it is most active in the brain. The general function of BACE1 is to form myelin sheaths for peripheral nerve cells. It is located on chromosome 11, and it is involved in the processing of amyloid precursor protein, better known as APP, to form amyloid beta. It is classified as a class 3 enzyme, which means it is a hydrolase. Its subclass is aspartic acid endopeptidases due to its breaking of peptide bonds. It does have a counter enzyme protein, which is BACE2. However, BACE2 is believed to be mostly astrocytic, which is a subtype of glial cells in the central nervous system, whereas BACE1 is more neuronal. In this next slide, we will talk about the structure of BACE1. The structure of BACE1 can be seen to have side chains of aspartic acid 32 and aspartic acid 228 in the active site. You can see from certain red arrows as the ones seen here that they indicate the displacement from inactive to active structures. The frontal view of the flap and the tyrosine 71 can be seen in structure C. And in structure D, you can notice the difference in location in the vicinity of the active site cleft and the flap between the active structure, which would be yellow, and the inactive structure, which would be blue. You can also see the hydrogen bonding networks by dotted lines, which can be seen right here. For the counter protein, which would be BACE2, there is not much information found because there hasn't been much research done on this enzyme. However, it is known that it is 75% homologous to BACE1, and it functions more as an alternative alpha secretase and as an antagonist to BACE1, as we have already said. It cleaves APP between phenyl 19 and 20 to abolish the alpha beta production, and it encodes an integral membrane glycoprotein that functions as an aspartic protease. So that's why it's allowed to cleave the amyloid precursor protein, and it's located as well in the brain and kidneys of mammals. In this next picture, we can see the comparison between BACE2 and BACE1. BACE2 is seen on the left side for both pictures, and BACE1 is seen on the right side. You can see in the labeled loop regions that they will adopt different conformations, which is the only main difference between the BACE1 and BACE2 structures, which is why they are 75% homologous. And now Kristy will talk to you about our research.
So our research looks at BACE1 and BACE2 and their enzymatic activities in Alzheimer's disease. So the BACE1 is the rate-limiting enzymatic activity, which is involved in the alpha beta or the amyloid beta generation. This is saying that Alzheimer's is is thought to occur because of the buildup of amyloid beta plaques that build up in the brain. These allow the brain to not function as well. We are going to look at BACE1 and BACE2, and this is the first time that they have been independently measured and are reported simultaneous activity of both their forms in Alzheimer's brains. So this is believed to be linked in the buildup of amyloid beta, which can be seen on the right, in brains of Alzheimer's patients, which is the main part of the plaques that build up. The amyloid beta is derived from the APP through cleavages from first beta, which is the BACE1, and then gamma, which is the C secretase. BACE1 first cleaves APP to release a large secreted derivative known as APP-S beta. This is then cleaved by the gamma, which produces the amyloid beta, which will then go on to produce the plaques. If the alpha secretase cuts APP first, it will create an APP-S alpha subunit, which is then cleaved by the gamma secretase, which produces the P3, which does not create the plaques that build up in the brain. So BACE1 is responsible for cleaving right above the ASP1 and the glutamine 11. BACE1 and 2 both compete for the same substrate and both cleave APP at the beta site. So next is the materials and methods. First, they collected their samples, which were frontal cortex samples of human brains, and kidney samples were isolated from rats. So the human brain samples were obtained from both males and females and were collected from the University of California and from the University of Kentucky. They had an additional 3 samples of late stage Alzheimer's that was collected from the UK, but this was only to help develop the assay, not in the actual analysis. So first with the samples, they homologized both of them and then centrifuged to create a pellet at the bottom. Activity of the enzymes BACE1 and BACE2 were in these tissue samples that they looked at. They then analyzed using Western blotting to determine where the BACE1 and BACE2 were active. They also used fluorescence to determine the activity and the ELISA. So the results for, they looked at the substrate concentration in brain and kidney samples and to determine the activity of BACE1 and 2 in the different tissue samples. So as you can see, BACE1 was much higher than BACE2 in the brain, and BACE1 was not present in the kidneys, at least their substrate was not present, which indicates that BACE1 is active in the brain and not in the kidneys, while BACE2 is active in the brain and very active in the kidneys. Next, they did a Western blotting, which was used to determine that BACE1 and BACE2 are both readily detectable in human brains. So as you can see, BACE1 is on the left and BACE2 is on the right. This is from the frontal cortex samples of the human brains, and as you can see, they did do BACE2 of the mouse kidney on the right, but not on the left for BACE1. This is the results of the ELISA. The star indicates that the results were significant, so as you can see, BACE1 was significant in both trials, while BACE2 was not significant. The lighter gray indicates the control, while the dark gray indicates the Alzheimer's patients that were looked at. So this shows that this is looking at the plaques, and this indicates that BACE1 is the main contributor of the plaques, which is produced by the amyloid beta on the brain. The next result is for the RIPA and the FA. The RIPA is when they use the soluble detergent, Well, the FA is when they use the insoluble fractions. BACE2 is not shown because there was no correlation, so they did not present the results. This shows a positive correlation between the amount of BACE1 and the amount of amyloid beta in the brain. So as you can see, both results are significant, which indicates that amyloid beta is present at high levels in Alzheimer's patients. For prediction, there is not much research going towards anything except for inhibiting BACE1. But by inhibiting BACE1, there was a study done by Steve M. Harrison on mice that showed physiological and biological differences when they did the knockout of BACE1. Because of this, they are not sure what will happen to humans. The other problem with treating Alzheimer's is the buildup of plaques is a long process. So to treat Alzheimer's patients, they would have to start treatment when the plaques form because there is no known treatment to get rid of the plaques. Right now, they are focusing on reducing the plaque buildup. So they would need an earlier way to indicate that there are plaques forming. So because of this, we thought one potential treatment would be to increase the amount of the alpha secretase because this will cut the APP at a different place and will stop out the amyloid beta units from forming and creating plaques on the brain. The one downside to this is there's not much research on the overexpression of P3, so we're not sure what would happen to individuals if alpha secretase increased production and the P3 was overexpressed.